visiting I2 and U of O this term as a sabbatical visitor. And Joanne is one of the few scientists that I know that's brave enough to study viral biodiversity in natural environments like soil, which I was told in grad school was not possible, but she is actually doing this, which we're going to hear about in just a moment. And I think in some ways she's uniquely trained to take on this big challenge. She has a PhD from the University of Colorado, where she worked with Noah Fear, one of the early pioneers in molecular microbial ecology. And she followed that up with a postdoc position with Matt Sullivan and Virginia Rich, University of Arizona, and then traveled with them to Ohio State, where she was trained in environmental virology. Think about it. And she was recruited by UC Davis in 2017 to start a program at UC Davis in environmental viromics. And she got tenure in 2023. Is that right?
Yes.
Yes, and is now an associate professor in the Department of Plant Pathology at UC Davis. And she's created this really productive and incredible program in environmental viromics at UC Davis. It's published dozens of papers and is supported by grants from DOE and USDA and NSF, including some really prestigious awards like the NSF Career Award and the Early Career Award from DOE, which are given to really promising scientists, and among the kind of the most prestigious awards that environmental scientists can receive early in their career. And one of the things I think sets apart her research and her program is that she's been trying to apply ideas and concepts from the general science of ecology to viruses of all things. She's going to tell us some about that today, as soon as I get out of the way. So please join me in welcoming Joanne Emerson.
All right. Thank you so much, Brendan, for that really, frankly, overly generous introduction. I hope I don't now disappoint you all, but yeah, no, it's a real pleasure to be here, and I want to thank especially Brendan and the CARBS Project for hosting me on sabbatical. And if any of this interests you, or if you're just curious about careers or anything else, and you want to chat sometime, I'm here until the end of March. So by all means, please feel free to get in touch. So yeah, without further ado, this is basically a talk about, what happens when you try to set up a lab to study soil viruses. And so we're going to call it Towards a Community Ecological Framework for Soil Viruses with a question mark, partly because I don't know if that's exactly where we're going, and it's certainly not where we intended to go. And it's also known as the magnificent, frustrating diversity of soil viruses. So these are a couple of postdocs in the lab who are at our primary field site near Davis. Luke seems to be thinking that this is very magnificent. Lucy thinks that it's frustrating. I think we've all had these sentiments about our research programs at one time or another. All right, so just by way of introduction, to get us all on the same page, some people think about soil a lot in this room, some people maybe not so much. Obviously, we walk on it, and there's all kinds of diversity in soil ecosystems. This is actually from an annual review in virology by Kurt Williamson, who is an awesome soil viral ecologist at William & Mary. And anyway, so this is just meant to highlight the diversity associated with maybe a root hair. And within this ecosystem, in addition to a bunch of microbial and quote unquote macrobial diversity, there are a bunch of viruses as well. And so the first set of introductions I'm going to do is introducing you to the soil environment and how it might apply to thinking about viruses, viral biogeography. So this is not necessarily something that I was thinking at the start of starting our program, but it's something that's become very important now that we're seeing data. And that's that differences in soil moisture content over both space and time, right? So within the same ecosystem over time, like in California, we have these long dry seasons and then rainy, wet winters. Around here, obviously, you have dynamics in moisture content over time, and then there are more arid regions and more tropical regions on the globe. And so all of these are going to have implications for habitat mixing and fragmentation and opportunities for interactions among microorganisms or compartmentalization. Thinking about soil relative to other ecosystems, it's a much more heterogeneous environment than some other ecosystems, for example, like the oceans. So there are differences in soil chemistry. You can think about this just kind of intuitively. Maybe you see a potting soil that's nice, organic, rich, very dark brown versus a sandy soil might be light in color. Those indicate huge differences in chemistry, which of course, at least partially contribute to differences in plant community composition. And so there's just a lot of heterogeneity, both locally and particularly globally, in terrestrial environments and different habitats. And again, in comparison to the ocean, it might be relatively well mixed. Although, of course, if you ask a microbial oceanographer, they'll tell you, oceans, they're also heterogeneous ecosystems. But I don't think there's a contest. And then if you're more interested in host-associated ecosystems, maybe the human gut is somewhere in between, give or take. All right. So I love the drama of this image. This is kind of an introduction to how I was thinking about soil viruses when the lab started. And so this was published in the New York Times a few years ago. So here's an E. coli cell. It is being lysed, broken open based on this viral infection. All these small light-colored particles are T4 bacteriophages that are causing the lysis of this cell. So this shows a couple of things. One, this microbe is very, very dead, obviously. So there's mortality involved in viral infections of single cells in the environment. You can imagine that this might lead to a chain reaction across a population, for instance, where these new viral particles are going to kill some other microbial cells, and you might have a change in community composition based on that. But actually, my favorite thing about this image is it highlights the change in chemistry that's occurring, right? So all of the nutrients that were, oh, this has died, that's fine, that were inside that microbial cell are now being released out into the environment, right? So that's a difference in the bioavailable compounds that were contained in the microbial cell versus were released. So that's sort of our interest in viral communities and viral ecology, although those are not the questions or things that we've been able to address to this point. It's been much more community ecology. So this is sweet. Thanks. Yeah. Yeah, so this is sort of like my excuse for not having a whole bunch of introduction on soil viruses. So there in theory weren't a whole lot of studies on soil viruses. This is not that there were zero. There were plenty. A lot of them were culture-based. In fact, Brendan's done a bunch of soil viral ecology. But when we were thinking more at the community scale, these are sort of some review or perspective articles that have been written over the last few years that suggest, at least among soil microbial ecologists and viral ecologists, maybe there was a lot of like hype and stoke, but not much data. So this is the excuse slide of like, why I'm not showing you a bunch of preliminary data. Sorry. Cool. All right, so now the meat of the talk. Here's where we're going. First, I'll catch us all up on the methods. How exactly are we analyzing these soil viral communities? And predominantly, this is basically how do we get from our soil sample to a viral species by sample matrix, essentially. And then magnificence and frustration, more or less in chronological order, give or take, of some of the stuff that we've been doing in the lab. So 3 very short studies that I'll introduce where we asked some questions that we didn't actually answer. We learned something else. And then we learned that we needed to really focus on spatial structuring. So we actually set up a study to study sort of distance decay relationships in viral communities. We realized we hadn't really done that over time. So then I'll introduce a study where we did those temporal successional patterns for viral communities. And then sort of at the very end, I'm going to provide what I hope is some hope for predictable interannual and global biogeographical patterns for soil viruses. So here we go, some methods. We focus very specifically on the viral size fraction. And so before I go into this, I just want to highlight for folks who think at all about amplicon sequencing data sets like 16S and ITS for assessing bacterial and fungal communities, there's unfortunately no such universal marker gene for viruses, so we have to use a shotgun metagenomic approach no matter what, meaning we're sequencing all of the DNA. But what we do before we do that is separate out the viral particles because they're smaller than everything else. So here's the sort of physical separation of viral particles. We start with a soil and buffer slurry. So the buffer has some chemistry that's going to facilitate removal of free viral particles from the soil matrix. Then we do a bunch of like fancy tedious lab work, a lot of shaking and centrifugation that ultimately will allow us to push that soil and buffer slurry through a 0.2 micron filter. And that 0.2 micron filter is mostly going to retain cells, larger organisms, that sort of stuff. And mostly what will go through are viral particles. There are some exceptions in both fractions, but there's a substantial enrichment just through physical separation of viral particles in that filtrate. And then those viral particles contain the viral genome, so we then can shotgun sequence the viral DNA and capture the viral community. So the next slide is going to be what are the bioinformatic approaches that we then use to figure out which viral species are in which samples and at what relative abundance. So first, yep, we've got our viruses. We're gonna, this is mostly based on short read Illumina sequencing. So we want to get bigger chunks of the viral genome. We do computational metagenomic assembly for that. Then we want to do a gut check and make sure what we have are actually viruses to the extent that we can be really confident in that. So we're going to look at the annotation of those longer pieces of viral genomes, look for hallmark genes like capsid proteins that are going to be involved in viral structural elements, and then say, okay, great, anything that is bigger than 10,000 base pairs and has these viral signatures, we're going to retain. And I'm going to show you some details on that. Quick sidebar, these are not perfect approaches. These are what the field has sort of come up with. I'm on board with it. I think this is the best that we can do. I'd be happy to argue with you day and night over methods, but hopefully don't get too tied up on whether you would want a slightly longer piece of the genome. We can talk about that over coffee. All right, so we're going to take our complete and partial viral genomes, 10 kb or larger. We're going to cluster them all at 95% average nucleotide identity into quote unquote viral species. And an important thing to highlight at this point is that, oh, thank you, this is a pointer now, is that we're clustering sort of at the data set level. So we have maybe, I don't know, 100 samples in a data set. Each individual sample is going to be taken to this point where we're reconstructing the viral genomes. And then we want a dereplicated set across our whole data set. This might be exclusively through this de novo approach where all the viruses that we're pulling out are from this sample. We could potentially also cluster with a publicly available database as well if we wanted to. So at this point, we're going to get a dereplicated set of viral species or VOTUs. And we want to know how abundant each is in each sample. I'm just schematically linearizing these genomes. We're going to get that abundance through a read mapping approach. Okay, so we're going to map the sequencing reads that came from each individual sample to our database, and we're going to get a feel for how abundant each virus is in each sample, mostly based on the coverage depth in this direction. That's about how many copies of the viral genome there were in that sample. But we also want a threshold in this direction to make sure we're confident in detection. So just one read at part of the genome wouldn't count. All right, next section is like a whole series of tiny little caveats that I feel like bother people when they're watching this talk and they come out at the end and they ask their exciting question and then they're crestfallen because like we just don't have the data to answer that question. So here are a few caveats right at the outset. The richness is thousands of viral species per sample, give or take, tens of thousands, thousands to tens of thousands. Only 1.5% of those are taxonomically classifiable at any level. Almost all the viruses that we're seeing, perhaps not surprisingly because nobody's really looked very hard before, are new. We're pretty sure they're viruses because of the approaches that we're using. We don't know what viruses they are, and so therefore we don't have any idea what hosts they infect in the environment. I would love to show you virus host like predator-prey dynamics, not going to happen yet, sorry. All right, those are methods. Now, moving on. This is the first of 3 little studies where we asked a question and answered a different one. So what we sought to do, this very first foray, it was a fun little road trip around Northern California with some folks in the lab. We thought that similar to, microbial communities, bacterial communities, viral communities would be different by habitat, right? So forest viral communities would be different from wetland viral communities, would be different from grasslands, regardless of whether the samples were collected next to each other or farther apart. Habitat would be the primary signal. This was not an epically perfect experimental design, technical replicates, biological replicates, just 30 soil viromes from Northern California. One point about this design, though, is that we did have what we thought were biological replicates within individual habitats. So of those 30, there were sort of 15 locations. Each location had a pair of samples. Now, the pair of samples might have been 40 meters apart, but it was still within the same instance of the habitat. So our viral community is structured by habitat. Let's walk through these data. So this is data from Devin Durham, who was an awesome undergrad in the lab, and Ella Shiretsky, who was a postdoc. We're looking at detection patterns of VOTUs across the 30 samples that we collected. So in those 30 samples, how many samples was an individual viral species detected in? And then how many viral species exhibited that detection pattern? So what you can see straight away is that most of the viral species were detected in a single sample. this isn't one special sample that was super diverse, it's any one of the 30 samples. And anybody who's attempted community ecology will know that if you're looking for patterns and your, species are detected in a single sample, that's not going to help you establish those patterns. So what we learned very quickly from this study was that we needed to start with less complexity. We need fewer habitats, more replication within the same habitat, and shorter distances between samples. All right, so we designed this study explicitly with that in mind. So this is Anneliek ter Horst, former PhD student in the lab, now Dr. ter Horst. So we went to Bodega Bay, which is just an atrociously hideous field site on the California coast. And so here, just very briefly, is the sort of set of locations where our samples were collected. We have 7 wetlands within one field site. So here's a 200-meter scale bar. It's a field site. It's not tiny, but it's relatively restricted. And then within each wetland, we collected a total of 9 samples. So 3 time points, triplicates per time point. We were very hopeful that with this sampling design, we could finally have sufficient data and replication for a robust cross-habitat comparison, at least at the local scale. Okay, here's what we found. I'll walk you through these graphs in case you're not familiar with the structure here. So this is an upset plot, and really you want to pay attention only to this first part of the plot. We're looking at the 7 wetlands. Each wetland had 9 samples. So if a viral species was detected in any one of those 9 samples, it would count as coming from that wetland. But there's not a single viral species in this sort of most abundant set of VOTUs that was detected in more than one wetland at this scale. The vast majority of viral species still were restricted to one of those 7 wetlands. However, at least now, instead of like only 10% or less being found in more than one sample in the previous study, including from the same habitat, right? Now more than 60% were found in more than one sample. They just mostly happened to be from the same wetland. So here's the fraction of VOTUs that exhibited different detection patterns in this data set. 40% were still found in one sample. So one of those 9 samples in one of the wetlands only. A little more than 50%, though, were at least found in more than one sample in the same wetland. So we're starting to make progress of seeing the same things again, slowly. But still only 10% were found in 2 or more wetlands, which makes it difficult to compare across habitats, different instances of the same habitat. We could still see a little hint of sort of habitat structuring biogeography here. And I'll walk you through this. It's a little bit of a convoluted mess, but what we're looking at are it's really 4 habitat types. It looks like 3. So these are halophilic habitats, so more saline. These are less saline, more fresh habitats. And then these were 2 habitats that happen to have a flip-flop between the saline chemistry that we measured and the plant communities that we expected to find there. So they either had a saline community with more fresh plants or vice versa. So these are actually not the same habitat, they're flipped. What we want to look at here is then a comparison of the circles and triangles. That would indicate that similar chemistry and plant community composition is showing similar viral communities. So these are sort of the same PICO-A plot. It's just that this is, this part redone. So you can see, first of all, that each of the 7 habitats are separating pretty well. But if you squint, you can see triangles versus circles, triangles versus circles. So there's at least a hint of separation driven by a few viral species that are shared and detected in the same instant, in different instances of the same habitat. Make sense? We're good so far? That was like we're good, like don't ask me a question. But if you have a question, you can ask anytime. All right, great. Okay, so 7 local habitats, too ambitious. Let's try one. Here's our 3rd study where we're asking a question that we didn't end up answering. So the question that we thought we were asking was, how do grassland viral communities compare under normal rainfall and drought conditions? This was led by awesome former postdoc, Christian Santos-Medellin, and it's in collaboration with a bunch of folks, including Jennifer Petridge at Lawrence Livermore National Lab and Mary Firestone at UC Berkeley, who had developed this awesome precipitation manipulation experiment whereby Some plots received 100% of annual rainfall on average over years. Some received 50%. I'm not even going to go into the EPIC experimental design because that's not really the signal that we saw in our data. So here's a bird's eye view of the plots that Chris has nicely color-coded by their location in the field. Here is what the viral communities look like in these samples at 2 different time points. We would have done more time points, but it was COVID. All right. So viral communities are very strongly spatially structured in this habitat, regardless of the precipitation manipulation treatment. You can see pretty clearly that the blue block is separating from the red block. And then within blocks, we have kind of this sort of almost distance decay in each. So clearly, there is spatial structuring of these viral communities in the field. And I'm not going to show you this next set of data, but I at least want to give you a hint that all is not lost. On PICO3, you actually do start to see an indication of the treatment effects on these communities. It's very convoluted. It would take me 7 slides to show you. So I'm not going to show you, but I'd be happy to talk about it later. But getting back to the spatial structuring, I'm going to show you the same data for the viral communities in a different way, and then the bacterial communities next to it from the same plots. So these are now our sort of distance decay slopes. Each point is a pair of samples. And on the x-axis for both, we have the pairwise distance between the locations from which those samples were collected. So just how far apart they were. And then on the y-axis, we have Bray-Curtis community similarity, either for the viruses here or for the bacteria through 16S rRNA gene amplicon sequencing over there. And you can see very quickly the y-axis scales, first of all, are the same. So the distance decay slopes for the viral communities are much steeper than for the bacterial communities. And this was very puzzling to us. It still is. I don't have an answer, but fortunately, Kyle Meyer with Brendan Bohannon wrote a great paper that Brendan just shared with me a couple of days ago that's actually really helpful for some of the things that we're going to look into for this. But if you have other ideas, definitely let me know at the end of the talk. All right. This is where we went, okay, fine. We probably need to deliberately sample over space and get a feel for what these patterns are rather than trying to measure something else and continually getting a signal that things are different over space. So this is PhD student Jane Fudima's ongoing work. She has 2 different grasslands where she's set up this spatial sampling design. So she has samples all the way down to the 10 centimeter scale where each of those corners is sort of 10 centimeters apart by edge. She has 6 of those. And then she has, all the way up to the field scale. So we're measuring over space deliberately. And I'll just show you a few pieces of information from her data set, but she's got all kinds of ongoing analyses. So first, let's look at an upset plot again at the 10 centimeter scale. All right. One example of those 6 replicated 10 centimeter squares, they all look approximately the same. Okay, good. Like, the biggest pattern is that these viruses were detected in all 4 samples, but all the rest of these are 70% of the data set. So 70% of the samples were not detected were not detected across the 4 samples over the centimeter scale. And so probably like the first thing you're thinking is the first thing we were thinking, which is like, okay, well, what does a technical replicate look like? Like probably this is just like natural variation, right? Our technical replicates we don't have from this site, but they're tight. They're at least 70%. Or more shared. So I think this is real biology or ecology, right? Great. All right, so what do the host prokaryotic communities look like in the same samples? Well, we were expecting them to look quite different given what we'd seen before. They are different, so 60%. These are metagenome assembled genomes. If you're not familiar with this approach, don't worry. It's just a different way of counting the bacterial species, quote unquote. So 60% of the mags were shared at that 10 centimeter distance, more than the viruses, but not 100% overlap that we might have expected. All right, so that's the small scale part of the study. I think, are we moving to the big scale? We are, okay. So these are just the viral communities. We're now looking at distance decay across the full scale of these 2 fields. We have a high precipitation grassland on the left and low precipitation on the right. Again, pairwise distance on the x-axis, viral community now dissimilarity on the y-axis. So obviously increasing dissimilarity over space, pretty striking. Once you get to about, 30 meters, give or take, you start to see an almost entirely different viral community. So that's pretty tough for trying to analyze any other pattern besides spatial distance in that field. Okay, so Brendan was here when this happened, and I think Matt was too. I asked my PhD student this morning, Jane, who's working on this, I was like, like we're doing this prokaryote versus virus distance decay comparison. Do you have these slides for, do you have the host data for this? Do you have the prokaryotes for this? And she was like, yes, I do. And then she texted me like in the text, she was like, do you want me to send like a nice pretty version of this? I was like, no, we're putting your text right up in my talk. So this was after this was already hooked up. This arrived on my phone. So here's a comparison of these distance decay curves for the viruses on top and the bacteria on the bottom. So you can see that the bacteria seem to top out at about 60% Bray-Curtis similarity, whereas the viruses get pretty darn different. So there we are. All right. So how are we ever going to measure soil viruses as a function of anything but space if this is all we're seeing in the field? Okay, fortunately, microcosms exist. So we, instead of trying to measure things in the field and dealing with all the heterogeneity there, we're instead taking soils from the field, homogenizing them in the lab, and making microcosms that are highly replicated. So here's just one example of a microcosm study that Chris did. And so this is, he actually went to 4 different grasslands around Northern California, and he homogenized within a grassland, so like sort of 4 different experiments of the same type of experiment, different source soil, and set up a whole bunch of microcosms that we could destructively sample over time from each. And so we took advantage of the annual wet up in California grasslands or in Mediterranean climates, where again, we have this seasonal drought, but then we have the first annual rains is known to facilitate an increase in microbial activity pretty much right away. And we figured probably if the viruses are going to be really active at any particular time, this would be a good time to capture the viral communities as well. So we have dry soil is the first time point, and then 24 hours after wet up with sterile water in the lab, all the way to 10 days. This is only a 10-day experiment. It's very short, 7 time points over 10 days. Importantly, 3 to 6 replicates per time point, because we had a feeling this was going to be a huge mess. So we wanted to see if we could reproduce what we were seeing. Super clean, actually. So this is an example from one of the 4 soils. All 4 did approximately the same thing, but each of the soils was completely different. So if you put them all in the same peak away plot, you just see almost 4 corners of the peak away plot. The biggest difference in viral community composition is between triangles and circles, which is the dry soil versus all of the wet soils at different time points. And then you can see clear successional dynamics over time from 24 hours to 10 days after wet up, from like the little rainbow colors. All right, what I really like is that this shows how tightly each of those replicates kind of looks, right? So like all 6 of the replicates here, then all 6 are changing in succession, whatever. So it looks like a pretty deterministic pattern, which is cool. But the one thing that you really can't see here is how much that viral community is changing. It's changing in the same way in each of our replicates, but is it pretty similar after 10 days, or is it really different? I'm setting this up because it's really different. So I'm going to show you the same data almost in a different way. This is, again, one soil. It doesn't include the dry soil. This is only wet soils. The X and Y axes are the same. So we have time since wet up, and these are pairwise viral community Bray-Curtis similarities. So for example, on the diagonal here are the the average pairwise similarities among replicates at the same time point. So you can see, as we saw on the last slide, that the replicates cluster pretty tightly. The viral communities remain pretty similar among replicates. But now if we look in the horizontal direction here, you can see pretty quickly viral communities are starting to differ over time. So 24 versus 48 hours, 24 hours versus 10 days post wet up. By the time you're at 10 days post wet up, you only have about 15% of the same viral species. But weirdly, the richness doesn't change much. So you still have thousands of viral species after 10 days. They're just thousands of different viral species than you had on day 1. And what does that mean? We'll find out. But again, now we're going to compare the viruses to the bacteria. Now we have all 4 soils in the different colors, pairwise temporal distance on the x-axis, and community similarity on the y-axis for the viruses on the left, and the bacteria, on the right. And I will say that we did this with both 16S and with these metagenome assembled genomes, and we see the same pattern where again, the viral communities are turning over much more rapidly in time than seem to the host bacterial communities from the same samples in all 4 grasslands. Still don't know quite why this is. Definitely crowdsourcing ideas. All right. So where do we go from here? We fortunately have this amazing field site near Davis where we think, well, we know because I'll show you the data in a minute, but we thought when we started it that we might have a chance of deconvoluting differences in habitat, space, and time at the same site. And so let me introduce you a little bit to this site over the next couple of slides. It's a grassland. Again, we have this like annual dry season for about 6 months of the year. So this was the end of the dry season in the field. We have our first annual rains. You start to have your grass growing and greening, yay. And then the cool thing about this site is that parts of it, like these are kind of a clay pan. So some parts of the site flood for some parts of the year, whereas these parts stay above ground or above the water table year round. So we sort of have 2 habitats within a habitat. We have these mounds and valleys, and I'll show you in the next slide a bit of a clearer picture of those. So this is a drone view. It's an oblique view, so this 5 meter, I made that up scale-wise, but it's approximately right. So this is during flowering where each of the mounds is in green and the valleys are in yellow. So what we did was we went to 4 mounds and 4 adjacent valleys where the valley was right next to one of the mounds. And we sampled every 1 to 3 weeks for about a year. And then we've continued to sample, but a bit less frequently for 4 years. So I'll show you, I think this is the next slide. We were waiting for sequencing data. And so Chris, the postdoc who was working on this project, was like kind of twiddling his thumbs. He was like, okay, well, why don't I just plot our viromic DNA extraction yields while we're waiting for sequencing data, see if anything comes out of it. We didn't expect anything. I'll show you in a second what we saw. But before I show you, it kind of is an implication of viral biomass, right? So like how much DNA you get out of your viral prep should be an indication of how many viruses are there. We just didn't expect to be able to resolve anything. But here's what Chris found. So on the x-axis is time, and we have viromic DNA yields, sort of quote unquote viral biomass on top, and soil moisture content on the bottom. This also matches with precipitation over the year. So you can see very clearly that when you have your first annual rains, over on the left side, you have a bloom in viral particles, so you have virus production. You have continued virus production or maintenance of the same viral particles throughout the growing season, and then when you have plant senescence and seasonal dry down, you see a decrease in viral particles, and then over the summer, you have very few to no viral particles. And then again, a bloom in the next rainy season. So that was cool to see, but it'd be nice to see the actual sequencing data too. So here we go. If we look at the point of the data that I'm showing you, we had, I think, about sequencing data to this point. We have a lot more now, but we don't have all the analyses done. So put all of the data from here into a PICO-A plot. What do you see? Well, the biggest separation is between mounds and valleys. So these are these 2 habitat features, sort of like sub-habitats within the same habitat, which is cool because, they're spatially farther apart, right? So your valley is right next to your mound in each case, but the mound that's farther away looks more similar than the valley that's right next door. But then, this is where it gets ridiculously cool. If you look within these habitat features, you actually see a slightly different pattern. So for the mounds tended to be, biogeographically more distinct by space. This isn't perfect. You'd love to see mound 1, 2, 3, 4 in the 4 corners of the plot, and we don't see that. I have no idea why these 2 particular mounds look more similar. We have not found anything that makes them actually seem more similar. But anyway, the main signal there is space rather than time, at least on the first 2 axes. Whereas for valleys, the signal is time rather than space. So this is spatially homogenized temporal succession. So you can see that, we have from December through May pretty clearly in the valleys. And again, the valleys, these are the habitats that flood ephemerally. They're not flooded year round, but they flood enough to hopefully make this a hint of dispersal limitation in the mounds and dispersal or mixing in the valleys. All right. So here's some other like little early results from the full 1.5 or a little less than 1.5 year data set. And I'll show you 4 years and some of that in a minute. But in addition to collecting those 4 mounds and valleys over time, Chris went and collected across the field from 24 different mounds. So this is like, it's quite an apples to oranges comparison. We don't know what temporal scale we should be comparing to what spatial scale, but essentially what we're looking at is an accumulation curve of the 24 mounds here. So that's the spatial accumulation curve, and the 4 different individual mounds over time, sort of the temporal accumulation curve. And accumulation curves are, as you increase your sampling effort, how many new species do you see that you hadn't seen in any of the previous samples? So you can see that diversity is increasing, or like sample diversity is increasing more steeply in the spatial, at the spatial scale rather than the temporal scale. And I would love to talk to you all about whether we should be considering different scales of space and time to make this comparison. But there you go. This was our first hint that there might be some recurring sort of like interannual or seasonal dynamics. So these are just the valleys, and we have time points sort of in their like little rainbow color, where these are the farthest apart in time for these. But if you're thinking about it, these are actually close seasonally to these in the subsequent year. So what you end up seeing is sort of like a successional pattern that sort of circularizes here, with the exception of these weird points that maybe those communities we didn't see in previous years. So my interpretation of this doesn't quite fit, so I could use some help, but my interpretation in theory is that these are communities that maybe we didn't see in previous years because we missed sampling on those days, right? Like we have this crazy temporal succession that's happening over like 10 days, but if we're sampling on 1 to 3 weeks, maybe we just missed these guys and we were lucky enough to capture these guys again. Except if the whole thing is doing this, shouldn't these still fall like vaguely somewhere over there? So I would love some help on that. And then this is cool. So this is Lucy Jaraska is a postdoc in the lab. She's now been following this work that Christian was doing. So Christian was doing this for like a year and a bit. Lucy's been continuing in subsequent years. So now we're seeing pairwise temporal distance in days over almost 4 years, with the dashed lines meaning, sort of when the year is starting again. And you can see pretty clearly some annual dynamics where communities both in the mounds and in the valleys are looking similar again after a year, and least similar after about 6 to 18 months. Kind of makes sense. All right. Oh yeah, okay, sorry, I should introduce this. This next thing is like totally off topic. Okay, so this is meant to be like, okay, this is what the like long-term temporal patterns are starting to look like, to the extent that we know what those are. So what's sort of the equivalent spatial scale? Well, the equivalent spatial scale might be the globe, give or take. So given that our viral communities are pretty different, 10 centimeters apart, very different across the same field, like tens of meters apart, probably on the global scale, you're just not going to see the same thing ever again, although this recurring dynamic suggests, I don't know, maybe. So let me explain what this data set actually is. So the Bodega Bay wetland data set that I introduced earlier in the talk, there were those 7 wetland habitats, 63 viromes. So what Anneliek did for this data set is she also took our giant database of over 500,000 viral like VOTUs that we have from around the globe. Most of them are from California because that's mostly where people are looking, but there are some from other locations. And so she mapped her sequencing reads back to this database to see where other VOTUs from her data set might have been detected. And so here's sort of the detection pattern. Obviously, most stuff is around California, where we've sampled other sites, but we do start to see the same quote unquote viral species at other locations around the globe. It's only 1.5% of all of the species in her data set, but we're starting to get there. And you can imagine that if things are pretty different over space and time within the same habitat, it's going to take a while before we sample the whole globe sufficiently to start seeing stuff more than 1.5% of the data set, but we'll get there. This is a little bit more encouraging. The next slide is actually from agricultural soils. This is from Russell Ranch near UC Davis. We just talked about Russell Ranch a minute ago. All right. Well, it was like an hour ago. But anyway, So this is, I haven't talked about any of our agricultural data sets. I'd be happy to if people are curious. But essentially, this was a particular tomato bulk soil and rhizosphere data set from near Davis, California. And Anna Lake did essentially the same thing, where she took all of the reads from that particular data set and mapped them back to her whole database of viral species from around the globe. And like, okay, not super surprisingly, most stuff is from California because that's where most people are looking at the moment. But you can see in agricultural soils in the browner colors and also rhizosphere soils in the greener colors. So there's sort of a habitat signature even in the few VOTUs that we can find around the globe. So that's more or less where we are over space and time at this point. So in summary, I just want to say that the emerging paradigm in soil viral ecology so far, very early days, is that these viral communities do seem to be pretty diverse and dynamic. They often differ over spatial distances of meters, maybe even centimeters, and temporal distances of days. We haven't really looked at hours, so I guess that's possible. But soil moisture seems to be a key driver of viral community composition and dynamics. That's both over time, right? So there's like, you know, boom and bust cycles with annual rains, plant growth, and dry down, but also biogeographically, there are patterns of, you know, mixing versus dispersal limitation that are coming out probably as a result of soil moisture content. And then I'm sure the ecologists in the room have like a legit word for this that probably involves some math. I'm just calling it rock, paper, scissors for habitat, space, and time. So when one of those things is sort of different enough to trigger a difference in biogeography, that's what you get. So when the habitats differ substantially, even on those local scales within the same sort of grassland ecosystem, habitat wins. That's going to be your strongest biogeographical pattern. Under dispersal limitation, particularly within the same habitat, space is going to win. You're going to get your spatial distance decay relationship. But when there's mixing, like when we homogenized our soil in our microcosm experiments or in the, you know, the valleys that ephemerally flood, time is going to win. You're going to have that sort of spatially uniform temporal succession. And then the last couple of slides is sort of, there might be still some hope yet for capturing the global soil virome, but it's a lot more complex than we anticipated. And these are the sheep that we encounter in the field sometimes. And there are a bunch of folks to acknowledge. I tried to highlight the leader of sort of each project as we went along, but in particular, data that you saw today were from Anneliek ter Horst and Jane Fujima, who were PhD students in the lab, Christian Santos Medellin, who's a former postdoc, Ella and Devin, postdoc and undergrad respectively, and Lucy, who is a relatively new postdoc, and definitely that grassland precipitation manipulation experiment. Want to acknowledge Mary Firestone from UC Berkeley and Jennifer Petridge from LLNL. Those were like massive efforts to set up an experiment that we found was just spatial structuring of viral community composition. And most of what you saw was DOE funded, a little bit NSF, and a little bit USDA. I'd be happy to take questions. Yeah, sorry, I don't, I just saw you first. Yeah.
This is super interesting. I'm really fascinated by this meta question of how you study or how you identify drivers, community dynamics, in communities that have such extreme temporal and spatial turnover. And I might have missed this, I stepped in a little late, but I was curious about like species interactions and all of this, just in the sense of my knowledge of viruses, they're mostly like obligate parasites. And you have this bacterial data. Did you notice, I guess, like correlations between individual taxa of viruses and bacteria would be tricky with that spatial turnover thing. But what about just like growth diversity? Like, is there a relationship between growth viral diversity and bacteria or other host organisms like plants or insects and like, do you have an intuition for which groups of organisms might be driving viral diversity or composition, right? In terms of these big categories of organisms.
Okay, I love this question. There are lots of pieces and I can pick, well, that means I can like cherry pick whichever ones I want. Okay, so a couple of things. First of all, I think these are, every single one of them, they're obligate host associated. In order to replicate, they require a host, and they're changing so much that the implication is they have to have encountered a host recently enough to have, made that change. So that's why the disconnect between the viruses and the host bacterial communities is so very puzzling, because there absolutely should be a match. So probably, well, there are lots of avenues to go down there, but let's say we found one particular group of viruses and hosts that we were interested in that we could look at. So with the precipitation manipulation experiment, some of the data that I did not show is that even though at the viral species level, we see those strong spatial patterns, if we're able to go up in taxonomic rank, which is super squishy for viruses, by the way, but like, quote unquote, if we're able to do that, then we can start to see some functional responses. So for groups of viruses that were predicted to infect actinobacteria, which tend to be drought responsive, those particular groups of viruses were found across the field in plots that were, well, it's not as clear as the drought manipulation, but in plots that had low moisture content at the time of sampling. So that's like a really roundabout way of not quite answering that question, because we don't have an answer yet. But the answer is we should be seeing patterns in virus host dynamics that we're not seeing. So we need to figure out how to measure those things in a way that will show us. I don't think it's going to be like magically the plant community composition is driving viral community composition over host bacteria. That doesn't like make sense to me. So yeah. Yeah. Yeah.
That the bacterial communities are really complex, really simple, and so would it be possible to with the right kind of radiate and knock down?
I like that idea, and we have, I wonder if we could go back into these data and look. We've done that, but not for that exact question, and I'll give you an answer that doesn't answer your question, but that's vaguely related. We had a greenhouse experiment where we compared a billion things because we didn't know which thing was going to be the signal, and one of the things that we compared was autoclaved soils versus conventionally managed soils versus organically managed soils and autoclaved versions of both. And it turned out that at the end of the experiment, the viral communities were distinct based on whether they were from organic or conventional management, which we expected, but separately, the conventionally and organically managed soils that had been autoclaved grouped together. So we think there was something that like dispersed in from the greenhouse environment to make that happen. So it's not at all the same as like distance decay and how that works, but there is something to what you're saying, and also the complexity. This is the hint that came from the paper that Kyle and Brendan had written, which is that absolutely one possibility is sort of a disconnect in the resolution that we're able to achieve in both of these communities. So how we fix that, we got to think pretty carefully about. Something that we had expected for a while, but it doesn't seem to be that it's the case, is that viral communities by nature, our approach captures very recently produced viral particles. That's just how it is. Like viral particles don't last very long in the environment, so you're capturing something that represents a short window of time. Whereas in soil is probably the best known place on Earth where you have, cells that are dormant, dead, active, free DNA floating around in the environment that is never going to be an organism again. So that could be part of the issue as well. Yeah. So suggestions for how to deal with that: crowdsourcing them. Great. Yeah. Oh, okay. So like, let me see if I understand this correctly. Like, if we had the most abundant VOTUs, would those ones be more likely to be found somewhere else on the globe? Unfortunately, no. And actually, I guess probably because we're sampling so deeply, the communities are pretty even. Like we don't have anything that's like really dominating, especially in natural environments. So how do we, okay. This doesn't answer your question, but it's interesting and related, I think. So for that spatial study well, it wasn't a spatial study, but we saw space as the signal in the grassland. Most of the VOTUs were not found on both sides of the field, but there were a few, I want to say like 20, 50, I don't know, that were found all across the field. And so it's definitely not exactly answering your question, but there was isolation by distance where those VOTUs that were shared looked sequence SNP-wise more similar with the stuff that was next to them than farther apart. So they weren't necessarily the most abundant, but they were like the most abundant VOTUs in a given sample, right? But they were common. That makes sense? Do you have any ideas about what like does yeah, okay. But yeah, I mean, like sort of stemming from that question, is there like a specific thing that you feel like you would want us to test like tomorrow, either with the data that we have or not? Great. Perfect. Yeah. Totally. Yeah. So, okay. So I'd say broad strokes, we have not personally collected bacterial biomass data, but yeah, like those, sort of boom and bust cycles throughout the California or Mediterranean grassland growing season, those are well known, and they match up pretty well with the times where the hosts are likely to be most active, times of viral production. So absolutely, I think that pretty clearly explains why you're not seeing very many viral particles over the growing season. But the problem is, where do they come from when the whole thing starts again if they're not surviving the growing season? So the population is surviving. Is the population surviving as like contingency, very low abundance, like viral particles, or is the population surviving as genomes inside host cells, that sort of thing? So yeah, for sure, I don't think the viruses are doing anything by themselves. Does that answer your question-ish? Yeah. Totally. Totally. Yeah. So that's exactly where we're going with, there's Lucy on this slide is focused exclusively on the dry season. Where the heck are the viruses in the dry season? And so yes, our predominant approach is only going to capture those free viral particles. So if we have a shotgun metagenome in tandem, we can look there, but unfortunately the like viral, the viral signal in those data sets is just like atrocious. So like we can't say anything definitively, but yeah, long story short, just sequencing the heck out of the dry season. And she also has some microcosm experiments where she's deliberately drying down and sort of tracking like in metatranscriptomes where stuff is maybe going. We don't have an answer yet, but we're attempting to figure out whether these things are inside host cells. And I will say that we don't have good evidence that they're integrated genomes because there's a specific suite of genes that's required for viruses to literally integrate into host genomes, and we don't see that suite of genes at high abundance. But there is like something called pseudo-lysogeny where you can have the genome inside the cell, but it's not integrated, and that's kind of what we think is going on. It's what we're looking for. Yeah.
And I was curious, from the microbiologist, who might know better communicates at all, whether like the introduction of water causes that same bacterial community to behave differently in a way that different viruses of like the drought rotation, and if you guys have any like ideas for how you guys are looking.
I love that point. I'd say like, yes, the communities definitely are behaving slightly, like you see successional patterns in the, bacteria as well, but they're more subtle because of the, differences in signal that we've discussed or whatever the case may be. But Oh, where was I going with that? Yes, like I think that has to be what's happening over time in those like short microcosm experiments, because it's not like there's dispersal as a possibility. It's just like the timing of when different viral populations are produced is changing. It has to be something related to host physiology. And we see like a little bit of a hint of that where like, you know, there's like a proteobacteria, actinobacteria switch, I think, where one is, I think that actinobacteria are dominant in drought and the proteobacteria come up after. I might have that backwards, but we see a suggestion that the viruses that are most abundant at those time points, to the extent that we can link viruses to hosts, follow those patterns. Yeah, but I think that's absolutely right. Great. All right. Thank you all.
Conversation, we're gonna have a few time, a few minutes.